Hello everybody and welcome back. So we are discussing module 5 on the biosynthesis of proteins, how proteins are actually synthesized in the biological cells. And we have seen that the synthesis of protein occurs in two consecutive steps. The first step was called transcription and the second one is translation. So, today we will discuss the second step, that is translation from mRNA to amino acids. In the process of transcription, in the last lecture I have talked about how the genetic information that is stored or that is encoded in the DNA is transferred to a sequence of RNA, and that RNA comes out of the cell nucleus. It comes out to the cytoplasm of the cell where the actual protein synthesis occurs, and DNA does not have to leave its place at the nucleus. And we have seen that how the exact sequence of mRNA is synthesized, taking the antisense strand as the template, and that actually makes exact replica of the sense strand. So, a sequence of the mRNA or the genetic information that is present in the newly synthesized mRNA is exactly a replica of the sequence of the sense strand that stays in the nucleus. So, that was about transcription. So, today we are going to see how the second step, translation, occurs. So, basically the synthesized mRNA to how it will be converted into a specific sequence of a protein. So, now why is it called translation? Why is the process named translation? The word translation usually means converting one language to the other, going from one language to the other language without changing its meaning. So, now I am talking English, which can be converted into other language such as Hindi, German, Japanese, so on. So, it can be translated to other languages. Same here. In this process, we will see how the language of mRNA is converted to the language of the protein. So, mRNA or RNA in general is nucleic acids, we know. So, it falls under the category of nucleic acids, and which is composed of the nucleotides, on the other hand, protein is composed of amino acids, and it contains peptide bonds. So, this process is all about converting the language that is encoded or encrypted in the form of nucleic acids to the form of amino acids or peptides. That is why this process is called translation. So, it uses the mRNA as the source of the code, and then that particular language is exactly converted to the proteins with a specific predetermined sequence. Specific predetermined sequence of protein. That is why it is called translation. And it all happens in the cytoplasm of the cell. Now, I am talking about the eukaryotic systems. So now what are the ingredients? So this process of translation actually happens with a chain of events or with a series of events, and there are few new molecules that we'll be talking about. What are the ingredients? First is of course the mRNA. That has been obtained from the transcription process, and this mRNA is messenger RNA, right? And this mRNA will act as our template. We'll use the information of the mRNA, and then the proteins would be synthesized. So this will be our template here. A second ingredient is called tRNA or transfer RNA. We'll see shortly how it looks like. What does it do? This actually carries the amino acids. So for the synthesis of protein, you need to bring the amino acids, multiple number of amino acids close together, and then only they can react to form the peptide bond. Transfer RNA is the molecule that actually brings or carries individual amino acids. And then the third ingredient and the most important of it. So once you have the other molecules, mRNA, tRNA and whatever else is required, all the ingredients need to be brought to a certain vessel where you can do the reaction. So you need container where all the ingredients can be put together, and then the reaction can happen. So in the cell, in this case, a ribosome is the vessel. Ribosome is the third ingredient that we need, and it works as a container or a working bench. On which protein synthesis takes place. We will shortly see what are the details of all these molecules. Ribosome is basically a group of proteins. Okay, so these are the key ingredients, or these are the key molecules that are involved in the process of translation. Now let's go one by one. mRNA you already know, but very briefly I will recap. It's a messenger RNA that was synthesized using the double helix, 5 prime to 3 prime, 3 prime to 5 prime, and this this we called as sense strand, this we called as antisense strand, antisense strand was used to synthesize RNA, messenger RNA. Antisense strand was used to synthesize a complementary sequence of RNA and in turn the sequence of this RNA would be exactly as that of the sense strand of the DNA, and then this mRNA comes out of the nucleus, five prime to 3 prime, and it is a RNA now with all the RNA nucleobases. Once it comes out of the nucleus, this is the nascent mRNA that was synthesized. Nascent means it actually contains all the code, or it is just a copy of the sense strand, full copy of the sense strand. So, nascent RNA, just born, just developed, but a large part of our gene is not important, to synthesize proteins. So, all the unnecessary part of the RNA would be removed, clipped off, and it will give you a matured mRNA. That is the transcription process. Now, there is one important thing I should mention about mRNA, is that if you write mRNA, now we will introduce a new term called codon. So, it contains the nucleobases, a group of 3 nucleobases or group of 3 nucleotides, RNA nucleotides present in mRNA is known as codon, and this a group of 3 nucleobases would be used for protein synthesis. They have some importance that we will see as we move on. So, mRNA can now be divided into a series of or a number of codons. This is one codon, this is another codon, this is another codon, this is another codon, this is another codon. So, all are codon, 3 nucleobases together. That is about the mRNA. And now comes the second one, tRNA, or known as transfer RNA, and this is how the molecule looks like. Large molecule, little bit complicated structure, but very important. So, if you see here, it is actually a single molecule. It starts with a 5 prime end, it is RNA of course, tRNA of course contain RNA nucleobases. It is a single molecule. Single RNA molecule, which basically means it is a single stranded compound that starts with a 5 prime end. I am sorry, here should be a phosphate at the 5 prime, goes all the way here, makes a different variations of structures that we will just talk about, but it without a gap, without a break, goes all the way up to the 3 prime end, and 3 prime end has a free hydroxyl group. So, it is basically a single molecule, single RNA molecule. RNA are usually single stranded, and this molecule is about 80 nucleobases long. That is the length of an typical tRNA. There are hundreds of tRNA molecules that are present in our biological systems, in our cell. So, it is about 18 nucleobases long. Now, there are many features that are associated with these structures, and some of them we will talk about. So, as you see here, it has this kind of structures, then again a bend here and a straight, then again a loop bend here, straight line, like this. It goes all the way there. So, in many places you can see it has a kind of a double stranded structure. This is double strand part, this is double strand region, this is double strand region. So, many double strand region are there, but this is a single molecule, which means partly self complementary in nature, and that actually forces the molecule to assume a double helix structure and a bend here, which is known as loop. You can call it loop, you can call it hairpin or something like that. So, it contains loop and stem. The double helix part or the double strand part is called as arm or a stem, stem as of a plant. Loop and stem structure, stem or arm, and there are 1, 2, 3, 4 of them. Now, how is this forming? If you have a sequence, maybe DNA, maybe RNA, let us fabricate a sequence G here, C here, T, C, maybe G, and then A, 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 and here. So, starting with this C, G, A, G, C. If you have a single stranded nucleic acid sequence, DNA or an RNA, for RNA it will be U, yes. If it is DNA it will be T. Okay, let us stay to the RNA itself, U. So, then it will be clear. Then if you look at this single strand, this part and this part are actually complementary. See, C, G, G, C, A, U, G, C, C, G, and they can form, because of the high GC rich content, they can form a pretty strong double bond. So, the molecule actually immediately restructure itself to assume this kind of conformation with this is G, this is C, C, G, U, A, C, G, and G, C, 5 prime, 3 prime. It's the same molecule, single molecule, just has bent itself, and in this loop there are all the A. So, this has a single stranded region, and this one is double strand region. This is known as loop, this is known as stem or arm. So, a single molecule can assume this kind of geometry or conformation. This is because of the strong self hybridization. This kind of structure is also known as hairpin structure. because it looks like a hairpin. Okay. So that's what happening here. A part of the sequences are self-complementary in nature that forces the molecule to bend here and assume the double helix. So the loop part are single stranded region, the stem part are the double stranded region. This, let us start with this the left side. This is of course the self hybridized, and then this region, this is called the D arm and loop. Their specific names have been given, the reason also. D arm and loop. This is the arm, this is the loop. D means dihydrouridine. In tRNA, we will see there are several number of modified nucleobases that are present. So, dihydrouridine is present instead of uracil or instead of uridine. Dihydrouridine is present in multiple numbers in this region, not all of them of course, but there are several of them are present in this part of the structure. That is why the name is D arm and loop. I will show you the structure of the okay, I will give you the details and then go to the structure. On the other hand, on the right hand side you can see this is termed as T psi C, arm and loop. Again T psi means pseudo uridine. Uridine should have been U here, but I do not know why T has been picked up. T psi is to denote the presence of pseudo uridine in this part of the structures, and similarly there is a modified cytosine here, usually 5-methyl cytosine M5C. 5-methyl cytosine is present here. In this part, this is called variable loop. Variable loop. And this contains a number of modified nucleobases, mostly 7-methyl guanine, as well as 2-methyl guanine. So, let us go to the structures again, and then we will come back again to the tRNA, because there are the actual functions or actual use of tRNA is not yet discussed. Why is it used for and how? So, here this is the ribose sugar. If there is a double bond here, this would be your uracil or uridine with the sugar. Without the double bond here, with H both, so 2H basically, This is saturated without the double bond. This is your dihydro uridine or dihydrouracil is multi, and that is present in I think 6 or 11, I forgot the exact number, depending upon the tRNA. Of course, that many number of modifications of dihydropyridine are present in tRNA in the D-arm loop. Now, what does it do? Why this is there? If you see, what is the difference between uridine and this? If you see, because you have lost the double bond here, this molecule become more flexible. More flexible than uracil. So, therefore, that helps adapt the loop and the stem structure. So, it makes it easy to mold things a little bit. That is why dihydrouridine was present here. Second is pseudouridine. So, this is your uracil or uridine. By the action of the enzyme pseudouridine synthase, is the enzyme that converts the uridine to the pseudouridine. See, here there is the double bond. Pseudouridine or termed as T-psi. So, what is the difference here? The difference is, see, this CN bond, there was CN bond in the uracil or uridine, sorry, not uracil, there was CN bond in the uridine, and this CN bond has been changed into a CC bond. So, this bond has been moved coupled with this carbon, and this carbon is your C5 carbon. So, it is actually, so this compound is actually your C5 ribose sugar instead of a CN bond. It has just been rotated, tilted a little bit, and this is the structure now with the NH free, this NH free. It changes the base pairing. So, what is the advantage? Of course, one advantage is that it changes the base pairing property now. Second is, you have formed a carbon-carbon bond. They have more rotational freedom. Free rotation around a carbon-carbon bond is possible. This is a single bond. Here the nitrogen has lone pair of electrons. So, it is a little bit more freely available for rotations. So, it can orient its geometry a little bit better. That is one. Second one is the base pairing. It changes its base pairing a little bit. So, this makes the compound more flexible, because you have to bend it to assume the loop and stem structures. So, that makes it little bit easier. So, this is about uridine, dihydrouridine and pseudouridine. If you write cytosine here, cytosine is double bond NH, no, CO here in double bond NH2, that would be your normal cytosine. Here there is a CH3 group, methyl group. This is your 5-methyl cytosine, or we write it in this fashion, M5C. 5-methyl cytosine is actually very common DNA modification that is present in our gene. It sometimes or quite often it protects our genome from attack by the external virus or other reagents. So 5-methyl cytosine is actually known as epigenetic tool, that you can see later on if we have the time to go. So this is about 5-methyl cytosine. It's a little bit modified version. Usually the cytosine doesn't have a methyl group here. Now the guanine part. So here is N, double bond, this is N, double bond, CO, NH, amine with a double bond, and there is nitrogen here. So this is your guanine, and this is the 9th position, you know. So 8, and this is the 7th position. So 7-methyl guanine would be methylation at the N7. This is the 7. Methylation at the N7 of guanine will give you the 7-methyl guanine. Guanine, or M for methylation, the 7th position of guanine. This methylation you have seen in some other places like during the DNA sequencing also. So this is one kind of modification. Second is 2-methyl guanine. All are in RNA framework. In NH2, NH, this is the second position, 2. Instead of NH2, it would be NHCH3. So this is your 2-methyl guanine, or M2G. So these are roughly, there are many other actually variations. These are roughly some of the modified nucleobases or the nucleic acids that are present in tRNA. Now coming back to the tRNA structures. This we have seen, this we have seen. Now here of course it has the partial double helix structure, and after the double helix is complete, then there is a hanging region. There is a single stranded dangling part of the RNA that is left over here, and that this is usually a 7 base pairs long, 7 bases long, this region. And at the end, the last 3 bases are always ACC. This is common to all tRNA. ACC, this sequence is common, it is fixed for all tRNA, and then at the 3' end you have a hydroxyl group, and this is where it reacts with the amino acid and attaches an amino acid in this position. That is how a tRNA would carry or would be able to carry a specific amino acid. So, that is one. This arm without the double helical part, of course, this is a single stranded arm, is known as the acceptor arm. Acceptor because it accepts the amino acids, and this is a recognition site. Actually, ACC is a recognition site. Another, so this part is very important for tRNA because it will be attached covalently with the amino acid which you would require for the peptide synthesis. Second, very important is this region. This is again a loop and stem structure. Here at the end, this is a 3 nucleobase set of 3 nucleobases. It is a 3 nucleobase sequence, and this is known as anticodon. Now you can guess what I am going to say. So, anticodon is actually complementary to the codon I have talked about in the mRNA. So, that mRNA has specific codons, many numbers, and tRNA has anticodons. If the anticodon is complementary to the codon, then that tRNA would hybridize to the mRNA, and that tRNA is of course carrying a specific amino acid. So, that is how the specific amino acid would be coming, would hybridize on top of a mRNA, and that is how the sequence of the mRNA is actually dictating which amino acid would come, because a specific amino acid has a specific anticodon. Okay, so that is about the tRNA. Now, let us look again. Here I have mentioned some of these features. So, tRNA 7 base pair acceptor, the sequence end sequence has to be CC. If you read 5' to 3' direction, it has to be CCA, and of course, there has to be free hydroxyl group, there has to be a free phosphate group, and this sequence is required for enzyme recognition. D arm is dihydrouridine here, T arm is pseudouridine, it is actually T psi, pseudouridine, and there are usually 6 number of pseudouridine present in this part, in the T psi and loop structure. The enzyme amino acyl tRNA synthetase, that actually will attach an amino acid here. I will show you the reaction to the tRNA. As I have said, this is a single stranded molecule containing about 80 nucleotides in the shape of a cover leaf. Looks like a cover leaf, isn't it? And this carries amino acids in the cytoplasm to ribosomes for protein assembly. We will see that later. 3 bases on tRNA here that are complementary to a codon on the mRNA are called the anticodons. So, this is your anticodon. For example, if the codon sequence in mRNA is UUA, then the anticodon would be AAU, of course, the complementary of it. So, basically in tRNA, the 2 parts are most important for its function. One is this, the other one is this. So, now let us look at what is the reaction that goes on, how the amino acid is attached to the tRNA. So, if you have, I am writing NH2 free form, of course it exists in zwitterionic form, but I am making it now simple. You have the amino acid. First of all, what happens, amino acid gets attached to an ATP. Here it is adenosine A, ribose, O, phosphate, phosphate, phosphate. So, this is your ATP. This is the reaction, it reacts here, diphosphate is eliminated. What you have now is A, O, phosphate, then O here, CO, this is your R, this is your NH2, your amino acid. So, this is your amino acid AA for amino acid. R is the side chain, it can be any amino acid. So, there is a acyl bond that has been formed. So, this compound is known as 5 prime because it is attaching at the 5 prime position. Amino acyl adenylate, 5 prime amino acyl adenylate, this moiety. And now you have the tRNA here, started with the 5 prime phosphate, goes here all the way to the free hydroxyl group. So, I am writing, so tRNA would also be attached with a ATP there. The last base of tRNA is A and OH. So, I need to draw that. So, tRNA it goes here and then last base is, here is the A, OH, here is the OH. So, this is the 3 prime hydroxyl group that reacts here, opens this up, and what you have is now tRNA this, here would be adenine, here would be OH, and there comes, in this position comes your the amino acid O, CO, NH2, and here is your R, this. So, with a acyl bond here, that is how the tRNA is attached with an amino acid. So, you have basically, if you see here, it will be OH, this OH would basically be attached like this, and this is the whole of this tRNA. That is how the amino acids, which we call AA now, would be attached with a tRNA. Now, as I have mentioned, there are hundreds of tRNAs present in the cell, each of them carrying a specific amino acid and each of them carrying a specific anticodon. So, whatever anticodon would be matched with the codon of the mRNA, that amino acid would be attached there. So, in the next lecture, we will see how the rest of the part goes on, how ribosome works, and then how actually the synthesis of the new peptide bonds happen. Thank you.